Let us look at the topic immobilization of enzymes and cell. So what is immobilization? We know that when we're talking about something mobile means something that can move, and immobile means something which is restricted in motion, which cannot move. So immobilization, when we are talking about the immobilization of enzymes and cells, that means we are making the cells or enzymes immobile. We are making them restricted. We are making them not able to move. Okay. So immobilization refers to the technique of confining or anchoring in our scenario, anchoring of what? Confining of anchoring of cells and enzyme on an inert support for their stability and functional reuse. Okay, so you're confining or anchoring the cells and enzyme, like you'll have to make them fixed onto an inert support such that they are not able to move. And why do you do this? For their function, maintaining their stability and for their functional reuse. So immobilized enzyme means an immobilized enzyme is defined as an isolated. You can isolate and purify the enzyme and then confine or localize it in a defined volume or space. An immobilized cell means you are confining or localizing the cells, high density cell, in a physical space. Okay, so you can do it in pellets, you can clump it or whatever, such that the movement is restricted when they are used. Okay, so they are also referred to as controlled biological catalyst. Okay, immobilized cells. So why do we need immobilization? We know that in a bioreactor, what do we add? We'll add the substrate, we'll add the microorganism, and the microorganism will use up the substrate, it will produce whatever product we need. And finally, after the process is completed, we will remove the microorganism. It can be bacteria, fungus, or whatever, and we will go for the downstream processing. But these microorganisms are actually active. Okay, like if there was some procedure whereby we can reuse these micro, we can collect them easily, then we could have reused them. Okay, so we are as the same stands for the enzyme. In some reactions, like you want to make glucose from starch, you don't have to add the complete microorganism. The only enzyme involved in the conversion of starch to glucose is amylase. So you can add your starch substances, okay, starch rich substrates, then you can add your enzyme amylase, you can allow the conversion to happen, and then after your conversion has happened, this enzyme is completely lost. So you don't want that. If there is some other method by which you can recover this, it would have been wonderful. And for this, we have developed immobilization. Okay. So when you're immobilizing the enzymes or cell, what can happen? You can easily recover the cells and enzymes from the media. Okay, because free enzymes in the media, they cannot be easily recovered and can only be used once. But immobilized enzymes or cells can be easily recovered and can be reused again and again. So that is the main aim for immobilizing your cell or your enzymes in a industrial bioreactor or a fermentation for using for fermentation. So there are different methods for immobilization. So looking at the different methods for immobilization of enzymes, IUPAC divides the immobilization techniques into four broad groups, that is covalent bonding, intermolecular cross-linking, adsorption, and entrapment. Okay, so covalent bonding means the enzyme is bound onto a water insoluble matrix via a covalent bond. There is a covalent bonding between the enzyme and the matrix. Intermolecular cross-linking means the enzyme molecules, okay, are intermolecular, they are cross-linked. Okay, intermolecular cross-linking of enzyme is taking place using some reagent which are multifunctional. some multifunctional cross-linking reagent. You are using a multifunctional cross-linking reagent so that there is a cross-linking happening between the enzyme. You'll see this in detail. Then adsorption means the enzymes are adsorbed onto the water insoluble matrix. There will be a water insoluble matrix on which the enzymes are adsorbed. Okay, this is a physical attachment phenomena. Then you have entrapment, where the enzyme molecules are trapped inside the water insoluble lattice or a semi-permeable membrane. So looking at the covalent linkage, here you can see the blue colored one is your enzyme and the silver colored or the gray colored sheet is the matrix. So the enzyme is linked to the matrix by the formation of covalent linkage. Okay, the covalent linkage is achieved between what? What of the enzyme? The enzyme will be having so many functional groups, and the functional group that is not essential for the catalytic activity of the enzyme will bind to the reactive group on the support system. So the reactive group will be present on the support system, and on the enzyme you will be having a functional group. These two will be reacting and forming a covalent bond such that the enzyme is fixed onto the support system. Okay, this is a widely used method, and the functional group that is used for the formation of covalent bond can be carboxyl group, hydroxyl group, imidazole group, indole group, phenolic group, etc. Okay, and the reaction involved between the enzyme and the support system, it can be acylation, amination, arylation, alkylation, etc. And which are the support system that can be used for covalent linkage? The support system can be, it can be agarose, cellulose, dextran, chitin starch, polyacrylamide, polyvinyl alcohol, it can be glass, it can be metal ides, bentonite, all these can be used as a support system. So you'll use a support system, and the support system will be having reactive groups. The enzyme will be there, the enzyme will be having functional group, and these two groups will form covalent bond such that your enzyme is fixed in place. Okay, but before you go for the enzyme reactive group interaction, the enzyme what the support interaction, the support group should be pre-treated such that it is able to bind to the enzyme. Okay. The pre-treatment step is very mandatory, but sometimes what happens is the enzyme cannot directly form bond with the support system. So in this cases, you will be adding some molecules like glutaraldehyde, which can form a linkage between the enzyme and the support. Glutaraldehyde will act as a linking substance between the enzyme and the support system. Then you have cross-linking. Here what is happening is there is no support system as such, but the enzymes, so many enzymes are cross-linked with each other. They are forming a network, okay, and cross-linking network like shown in the figure. So the enzyme will react with the, it will interact with the next enzyme that will interact with so many enzymes, and this will form a cross-linking structure. Okay, so in this case, you will have to use something called a cross-linking agent. The cross-linking agent will be added with the enzyme such that these agents which have so many active site, okay, this cross-linking agent will be having so many active site. So each active site will bind to one or two enzymes such that these enzymes, so many enzymes are cross-linked together. One very good cross-linking agent is glutaraldehyde, okay, which this has two cross-linking sites. Okay, this cross-linking is a very strong bond formation, and this is irreversible. It is resistant to high pH and temperature. So sometimes the cross-linking might result in the loss of function of the enzyme and in the denaturation of enzyme. Next one is adsorption. This is the third method. Okay, adsorption is a physical interaction of the enzyme onto a water insoluble matrix. So here a physical bonding is formed on the surface of an inert support. and the interaction between the enzyme and the support will be very weak, like van der Waals interaction or hydrogen bond formation, and hence this is not very stable. So this is a simple and inexpensive method, and how you do this is you will bring together, okay, the enzyme solution and the water insoluble matrix solution together. And what happens, the enzyme will come and bind onto the matrix. You will wash off all the unbound or unadsorbed, here sorry, it is not bound, it is unadsorbed enzyme which has not been adsorbed. Okay, so how the extent of adsorption, how much adsorption is done depends on many factors, like the nature of the support system you are using, the nature of the uh, inert support system you are using, the pH, the temperature, the time you are giving for the interaction, the enzyme concentration that has been used. All this determines how well the adsorption is done. So what are the different supports you can use? You can use alumina, bentonite, calcium carbonate, calcium phosphate, cellulose, charcoal, clay, resins can be used, Cefadex, silica gel, all these can be used to adsorb the enzymes. So you're absorbing the enzyme onto the support system. The support system has the enzyme fixed onto it. You can use this in the bioreactor. The reaction can continue. After the reaction is there, you can take out the support system. You can separate the support system or the adsorbed enzyme from the bioreactor easily by some techniques. But here there is a chance of washing off of enzyme because the interaction is very weak. There is a chance that the enzyme may wash out of the material. But the enzyme, since the interaction is weak, again the enzyme need not be inactivated. Then the enzyme will be active. There is no decrease in activity of the enzyme due to adsorption. This is the next technique. It is called as entrapment. You are trapping your enzyme inside a matrix like this. Okay, you can trap it inside a matrix or inside a droplet. Okay, but here what is there, the enzyme is just trapped inside the structure, but it is not having any chemical reaction happening between the support and the enzyme. Okay, here a cross-linking polymeric network is formed around the enzyme. Okay, and the enzyme is physically placed inside this network. Polyacrylamide for cross-linking purpose or entrapment purpose, you can use polyacrylamide gel, you can use starch, collagen, gelatin, etc. Silica gel is also used for entrapment. The size, the pore size of this entrapped matrix should be such that the enzyme cannot escape out of the matrix. Okay, the pore size should be smaller than the enzyme, but if the reaction has to happen, the substrate should meet the enzyme and the product should be formed. Okay, so the pore size should be such that the enzyme is not able to come out of it, but the substrate can come into the entrapped structure. It can react with the enzyme, it can form the product, and the product can escape out of the structure. So enzyme cannot move, but the substrate and product can move in and out. Okay, but some cases, if the pore size are slightly bigger, the enzyme might leak out of the network. Then you have microencapsulation, which is a type of entrapment technique. Okay, here what is happening is small, very small, and beads are formed. Microencapsulation consists of packaging the enzyme inside spherical beads or spherical capsules, and the size of this capsule will be 5 to 300 micrometer. So that is why it is called as microencapsulation, very small size. And these beads can be permanent structure. The encapsulation structure can be permanent or a non-permanent liquid structure. But these are usually semi-permeable such that it will allow the entry and exit of substrate and product. So it can be made up of cellulose, nitrate, polystyrene, etc. And the advantage of this microencapsulation is because of its small size, when you're considering in a particular fixed volume of space, so many small microbeads When you're comparing it with larger size microbeads, the smaller size microbead will be having like more surface area. Okay, and this increase in surface area ultimately lead to the increase in the enzyme substrate reaction. So the reaction whatsoever can occur very fast. Okay, but the disadvantage, you also have a disadvantage is that for microencapsulation, you need high concentration of enzyme and only very low molecular weight substances can pass through this microencapsule. So you can't use this widely for all enzyme substrate reactions. Okay, these are the figures of adsorption, microencapsulation, cross-linking, entrapment, and covalent attachment. So these are some applications. So we have the third example, amylase. We have discussed that starch can be produced from glucose, and for this production you can use entrapped or immobilized amylase enzyme. For production of L-amino acid, you can use immobilized amino acylase enzyme, and glucose isomerase enzyme can be used for the production of high fructose syrup. So what are the advantages of immobilized enzyme? They can be easily separated from the reaction mix. Since the enzymes are immobilized, they can be easily separated and they can be reused. The immobilized enzymes are stable over the broad range of pH and temperature when compared to non-immobilized enzyme, and the enzymes will not be present in the waste stream. Whatever the waste you're letting out of a bioreactor, it won't have any enzyme. So the denature of enzyme, that step can be omitted. The immobilized system can be, the enzyme can be continuously used. Okay, so this is a continuous process, reusing of the enzyme, and hence reduced costs in industrial production can be there. You can reuse the enzyme so many times, so it is cost effective. Okay, you can control the catalytic effects rather than free enzyme. The immobilized enzymes are more stable. Okay, there is a ease of new application for industrial and medical purpose. You can easily use this. The immobilized enzyme permit the use of enzyme from organisms which do not normally be safe. There are some organisms which are not safe, which are considered as a hazard. Okay, even the enzyme from these organisms can be extracted and safely used. That is a very big advantage. If you were going for the organism as such, this would have been a issue of biosafety. But since you are taking out the enzyme and immobilizing and using it, it is okay. Then normally immobilized enzymes are preferred over immobilized cell because the enzymes are specific. Okay, due to the specificity of the enzyme, whatever reaction you want to carry out, the enzyme specific for that reaction can be added. Okay, and hence immobilized enzymes are preferred. Now let us see the immobilization of cell. Okay, so immobilization of cell can be done when the process involved is a multi-enzyme reaction and or involves cofactor. This means you are not relying on a single reaction, you are relying on the use of different enzyme substrate reaction. Okay, the bacteria or the fungus that is used will be catalyzing so many reactions, then you can't the immobilization and use of a single enzyme won't suffice, and sometimes for the activity of the enzyme it might need a cofactor. Okay, so this cofactor is present inside the bacterial cell. So in such cases, you can add cofactor into the bioreactor, but still, in such cases, immobilization of cell can be done. Okay, prokaryotic cells are mainly entrapped because they are easy to use and they have a faster metabolism. But for the production of antibodies, immunoglobulin, and complex molecule, immobilized eukaryotic cells have also been used. Okay, the two techniques for the immobilization of cells are entrapment and surface attachment adsorption. So you can look at this in the previous slide. What is the technique? Entrapment, microencapsulation, and surface attachment can be used. So these are the application. You can use immobilized E. coli for the synthesis of aspartic acid. Okay. Then E. coli can again be used for the synthesis of production of L-tryptophan from indole and serine. Okay. Saccharomyces cerevisiae can be used for the hydrolysis of sucrose. You can use immobilized. This biocatalyst can be immobilized and used. So these, because these are involved in more than one enzyme reactions. So what are the advantages of immobilized cell? The immobilized, we have seen what are the techniques for immobilized cell. Those are two techniques mainly used. Why immobilization of cell? Why? What are the advantage? The cell, when the cells are immobilized, the reaction is more homogeneous than using free cells in a bioreactor. Okay. The lag period which occurs in a batch fermentation is eliminated. Okay. And the accumulation of product is associated with the non-growth phase of the cell. So what happens here is, when you're immobilizing your cell, you will be growing your cell, you'll be growing it in the up to the logarithmic phase, and these actively growing cells will be immobilized. So inside the bioreactor, you can bypass the time taken for the log phase. So there is no time. So all the time you are having a product being formed. There is no time lag for the growth of the microorganism. Okay, so it is more feasible to run immobilized cell continuously as high dilution rate. So even though the number of cells required for a bioreaction is very low, you just need a very diluted cell. Okay, you can easily use immobilized cell. If it was free cells, okay, a non-immobilized bacterial or fungal cell, when you're using a very low concentration of cell, there is a chance of washing out. But in case of immobilized cell, even though the concentration is low, okay, there is no washing out, there is no risk of washing out. Okay, immobilized cell, even though initial cost for using immobilized cells are high, okay, it is finally, when you are taking everything into consideration, it can be cheaper than the normal batch culture because you are saving time here. You can reuse the cell. So the initial capital cost will be high. Okay, you'll have to prepare your immobilized cell. You'll have to grow the cell in a separate bioreactor. For that, the initial capital cost will be high. The use of immobilized cell eliminate the need for, so when you're using the immobilized enzyme, you will have to extract the enzyme, you'll have to purify the enzyme. Okay, so these steps can be bypassed by the use of immobilized cell. When compared to the immobilized enzyme, that is the advantage of immobilized cells. And one more thing, sometimes as we have talked earlier, multi-enzyme reaction need to be happening inside a bioreactor. In such cases also, immobilized enzyme cannot do the work. You might need more than one immobilized enzyme inside a bioreactor. So it is easier to use immobilized cell for this case. Then immobilized cells are more suited for multi-step process, and cofactor regeneration is not a problem. If it was immobilized enzyme, you might have to keep on adding cofactor, but here, since it is a cell, the cofactor whatsoever is needed will be present inside the cell. So these are the advantages of immobilized cell.